LRP5 mutation. LRP5 is a gene that controls bone density, and a specific mutation in it can make your bones virtually unbreakable. While human bones are normally somewhat porous and vulnerable under high impact, people with this rare variation possess skeletal structures so dense that standard medical saws struggle to cut through them. Severe vehicle collisions and intense sports falls that would easily shatter an average person's femur result in little more than minor bruising. The mutation causes an overactive signaling pathway that constantly tells bone cells to deposit minerals without stopping. But it comes with a silent drawback. The sheer weight of such dense bones makes floating or swimming in water almost impossible, turning the human body into a heavy anchor. Furthermore, excess bone growth inside the skull can pinch cranial nerves, leading to potential hearing loss or facial paralysis later in life. DEC2 mutation. Known as the short sleeper gene, a single point mutation in DEC2 allows a tiny fraction of the global population to thrive on just 4 hours of sleep at night. While the average person suffers brain fog, mood swings, and severe health issues from chronic sleep deprivation, natural short sleepers wake up fully refreshed, alert, and energized after half the normal rest. Their brains clear out toxic metabolic waste faster and consolidate memories far more efficiently during short sleep cycles. They gain up to 3 extra awake hours every single day, giving them thousands of bonus waking hours over a lifetime. However, because their nervous systems are constantly running on high efficiency, they often display intense, restless drives and struggle to remain still during periods of forced inactivity. PCSK9 mutation. This rare genetic anomaly acts as a natural shield against cardiovascular disease by lowering bad LDL cholesterol to ridiculously low levels. Under normal conditions, the PCSK9 protein destroys receptors on the liver that clear bad cholesterol from the bloodstream. When this gene is mutated or disabled, those receptors remain permanently active, wiping out LDL before it ever gets the chance to build up inside arterial walls. People carrying this mutation can eat high-fat diets and maintain arteries as clear as a newborn baby's, reducing their lifetime risk of heart attacks by up to 88%. Pharmaceutical companies spent billions studying this tiny genetic glitch to develop modern cholesterol-lowering drugs, but those born with it possess a natural biological defense that lasts forever. Tetrachromacy. Most humans are trichromates, possessing 3 types of cone cells in their eyes to perceive roughly 1 million distinct shades of color. But due to a rare mutation on the X chromosome, a small percentage of women develop a 4th distinct cone cell, granting them full tetrachromacy. Instead of seeing a simple blue sky or green leaf, a tetrachromate perceives subtle gradations, tones, and dimensions invisible to the rest of the world. They can distinguish between over 100 million different colors, noticing tiny shifts in light and pigment that look completely identical to everyone else. The challenge is that the modern human world isn't designed for 4-dimensional color vision. Artificial lighting, digital screens, and manufactured dyes often look harsh, unnatural, or disorienting to them because technology is engineered for basic 3-cone perception. ACTN3 gene mutation. The ACTN3 gene produces a protein called alpha-actinin 3, which drives explosive high-speed contractions in fast-twitch muscle fibers. If you possess the functional version of this gene, your muscles generate immense power instantly, making you naturally built for sprinting, weightlifting, and combat sports. Almost every single elite 100-meter sprinter carries at least one functional copy of this protein. On the flip side, roughly 18% of the world's population completely lacks functional alpha-actinin 3 due to a double mutation. While this absence makes rapid acceleration harder, it grants superior energy efficiency during long-distance endurance running, proving how ancient human evolution traded short bursts of speed for relentless stamina. ABCC11 mutation. A single letter change in the ABCC11 gene determines whether your body produces sweat that smells terrible or stays completely odorless. The non-functional version of this gene stops the secretion of specific lipids and proteins in the apocrine sweat glands, which are the primary food source for odor-causing bacteria under your armpits. If you carry 2 copies of this mutated gene, your sweat contains virtually no compounds for bacteria to break down, meaning you never develop underarm body odor even after heavy physical exertion.
As an added biological side effect, the same genetic variant changes your earwax from wet and sticky to dry and flaky. It is exceptionally common in East Asian populations, but remains extremely rare among people of European or African descent. FAAH-1 mutation. Known as the high-altitude gene, this specific genetic tweak was inherited thousands of years ago from interbreeding with ancient Denisovans. In normal human bodies, entering thin, high-altitude air triggers a massive surge in red blood cell production, which thickens the blood, causes severe altitude sickness, and dramatically increases the risk of stroke. The FAAH-1 variation found in native Tibetan populations prevents this dangerous overproduction. Instead of pumping out excess red blood cells that sludge up the cardiovascular system, their bodies adapt by widening blood vessels, increasing nitric oxide production, and maintaining smooth, efficient oxygen delivery to vital tissues. It allows them to run, work, and thrive at elevations above 14,000 feet without gasping for breath or suffering long-term organ damage. Myostatin mutation. Myostatin is a protein that acts as an internal biological brake, stopping human muscles from growing too large and burning excessive metabolic calories. When the gene responsible for myostatin is mutated or disabled, this muscle growth limit completely disappears. Individuals born with myostatin deficiency develop twice the average muscle mass and significantly higher strength without putting in extra effort at the gym. Their bodies continuously build dense, sculpted muscle while burning off subcutaneous fat at an accelerated rate, leaving them looking like professional athletes even in early childhood. While it provides incredible physical advantages, the massive calorie requirement of maintaining such heavy muscle tissue means the body demands vast amounts of fuel just to function day to day. FAAH-OUT mutation. Discovered in a Scottish woman who reached her 60s without ever experiencing physical pain, chronic anxiety, or fear, this ultra-rare mutation disables the enzyme that breaks down anandamide, the brain's natural bliss molecule. With anandamide flooding her nervous system constantly, severe burns, surgical incisions, and bone fractures feel like nothing more than mild pressure. Cuts and wounds heal at remarkable speeds, with minimal scarring. Mentally, the brain remains completely calm during high-stress panics, making worry or psychological trauma virtually impossible. However, the loss of physical pain deprives the nervous system of its primary warning mechanism, meaning severe internal injuries or infections can go completely unnoticed until they become life-threatening. SCN9A mutation. Unlike the soothing bliss of the FAAH-OUT mutation, a total loss of function mutation in the SCN9A gene shuts down voltage-gated sodium channels in pain-sensing nerves entirely. People born with this condition feel zero physical pain across their entire bodies from the moment of birth. They can step on broken glass, touch burning stoves, or break joints without shedding a single tear or registering discomfort. While it sounds like a real-life superpower, it is one of the most dangerous genetic traits in existence. Toddlers with this mutation frequently bite off parts of their own tongue or lips while teething, and adults can suffer from silent appendix ruptures or internal bleeding without ever knowing anything is wrong. Most require constant medical screening just to survive childhood. ALDH2 deficiency. A widespread mutation among East Asian populations alters the ALDH2 gene, which produces the enzyme responsible for breaking down acetaldehyde, a toxic byproduct of alcohol metabolism. In people with this genetic variant, acetaldehyde accumulates in the liver and bloodstream at concentrations up to 20 times higher than normal. Within minutes of taking a single sip of alcohol, the facial skin turns bright red, the heart starts pounding violently, and intense nausea and headaches set in. It's the body's emergency alarm system going off. While this mutation acts as a natural deterrent against alcoholism, continuing to drink despite the reaction dramatically increases the long-term risk of esophageal and stomach cancers due to chronic toxin exposure. Golden blood phenotype. Blood types are determined by antigens on red blood cells, with most people having varying combinations of the Rh system's dozens of antigens. Rh null, known informally as golden blood, occurs when a person lacks every single one of the 61 antigens in the Rh system due to rare mutations in the RHAG gene. It is so exceptionally rare that fewer than 50 people on the entire planet have ever been identified with it. Golden blood is the ultimate universal donor for anyone with rare Rh blood types, making it priceless to medical science. However, living with golden blood is extremely dangerous because no other blood type is compatible with theirs. Receiving a blood transfusion from a standard donor would cause a catastrophic fatal immune reaction, leaving carriers dependent on a tiny international network of donors. CCR5 delta 32 mutation. The CCR5 gene codes for a protein receptor on the surface of white blood cells, which acts as the main doorway for the HIV virus to enter and infect the human immune system. A specific 32 base pair deletion in this gene distorts the shape of the receptor, locking the biological door completely shut. Individuals who inherit 2 copies of this mutated gene are almost entirely immune to HIV infection, as the virus simply cannot latch on to their immune cells. Scientists believe this mutation became more common in northern European populations due to historic natural selection during deadly plague epidemics. Today, genetic therapies are using bone marrow transplants from CCR5 delta 32 donors to successfully cure HIV in medical breakthroughs.